Welcome, curious minds, to the deep dive. Today, we're embarking on a truly fascinating journey, one that requires us to shrink our perspective right down to the cellular level. We're going to explore the most fundamental processes of life itself, and in doing so, uncover something, well, pretty remarkable.
Which is?
How these tiny, intricate mechanisms profoundly dictate the very pace of our aging.
Indeed. And, to really grasp this, let's start with a little thought experiment.
Okay.
Imagine just for a moment your own home, or maybe your workspace. What happens if the trash cans are constantly overflowing?
Right. Gets messy fast.
Exactly. Or if things break down and you just, leave them unrepaired.
Yeah.
Or maybe you stop bothering to sort and recycle valuable materials.
Yeah, total chaos eventually. Disorganized, inefficient.
Unlivable, perhaps.
Yeah.
But take that exact concept, that really vital need for continuous maintenance, and scale it down, way down.
To the most fundamental unit of life, the cell.
Precisely.
That's a perfect analogy, I think, because just like your home, cells absolutely need their own intricate, highly specialized cleanup crew. Our focus today is squarely on two incredibly powerful cellular quality control mechanisms. One's called proteostasis,
Right.
and more specifically, a process known as macroautophagy.
Macroautophagy, yeah. We'll dig into that one.
And we're going to unpack how their efficiency isn't just about, mere cellular tidiness. It profoundly influences the entire complex process of aging, whether our cells thrive or, falter.
So our core mission in this deep dive is to meticulously illustrate precisely how cells maintain their proteins and organelles in optimal working order, how they function at peak efficiency.
Okay.
We'll be zeroing in on the specific, sophisticated tools in their arsenal. Things like molecular chaperones.
The guides?
Kind of, yeah. And the ubiquitin proteasome system, which we'll probably just call the UPS for short.
Okay, UPS. Got it.
And then the multifaceted autophagy lysosome pathways. These are basically the cellular engineers, the waste managers, and importantly, the recycling specialists.
And just to set clear expectations for this particular deep dive, while there are definitely other vital cellular processes that contribute significantly to aging.
Oh, absolutely. Like mitochondria.
Exactly. The powerhouses of the cell or the broader concept of cellular energetics, we won't be diving deep into those today.
Right. Focus is key.
Think of them as equally fascinating topics, maybe for a future deep dive. For now, we're concentrating intensely on how cells precisely handle their internal waste, how they maintain order, and crucially, how they recycle their components.
So the central overarching question we're really tackling today is this. How do cells, on a continuous basis, manage to prevent a debilitating buildup of what we might just call cellular junk?
Junk like damaged proteins, worn-out organelle.
Exactly. Stuff that could quite literally clog up their vital functions and bring everything to a grinding halt. What are their strategies for maintaining, molecular integrity?
And our overarching goal here, to vividly illustrate how the cell's clearance capacity, its innate fundamental ability to efficiently dispose of and recycle waste, is a truly critical, maybe even overriding determinant.
A determinant of what exactly?
Of the rate of cellular failure. And this isn't just about the natural process of aging. It also plays a profound role in the progression of some really devastating diseases.
So we'll paint a picture.
Yeah, a stark, almost cinematic picture, contrasting the grim scenario we see in severe neurodegeneration, where the cellular clearance fails catastrophically.
Like Alzheimer's or Parkinson's.
Precisely. Contrasting that with the remarkable resilience and vitality seen in, say, a healthy centenarian, someone where this quality control evidently prevails, allowing them to thrive against the odds.
So it's a compelling tale of two extremes.
Exactly. Fundamentally hinged on how well our cells manage to clean house.
Okay, that's a great setup. Where do we start?
All right, let's unpack this and begin with what's known as the Proteostasis Network, Cellular Quality Control 101. It might sound like a dense technical term.
It does sound a bit technical, yeah.
But the underlying concept, when you really break it down, is truly elegant, almost beautiful in its simplicity.
Precisely. At its core, proteostasis refers to the homeostasis of the proteome.
Okay, let's break that down. Proteome.
The proteome is the entire dynamic collection of all proteins within a single cell at any given moment.
Yep.
It's constantly changing.
Got it. So proteostasis isn't just about having proteins.
No, not at all. It's about maintaining those proteins in precisely the right shape, the right location within the cell, and crucially, in the right amounts.
All three are important.
Absolutely. It's all about keeping things optimal for the cell to function properly, performing all its thousands of tasks. Think of it as maintaining protein equilibrium.
That's a powerful distinction. So if the proteome is all the proteins, then the proteostasis network is like the cell's highly sophisticated quality control department.
That's a great way to put it. Yeah, a quality control department operating relentlessly 24/7.
Tirelessly working to ensure every single protein molecule is perfectly functional and properly managed.
It's a constant dynamic process. It involves this delicate dance of creating new proteins, guiding them to fold correctly.
Which sounds tricky.
It is. And then attempting to repair them if they become damaged. And if all else fails, efficiently getting rid of them before they cause any harm.
So it's creation, guidance, repair, and disposal.
Exactly. It's truly a complex system, always adapting to the cell's needs. And this incredible continuous feed of molecular management is achieved by what's collectively known as the proteostasis network, or PN.
The PN.
And this network isn't just one thing. It comprises several key components working together, like a highly integrated system.
Let's start with the first, maybe the most foundational component.
Good idea. Let's talk about molecular chaperones, the protein architects and first responders.
Architects and first responders, I like that.
They really are crucial assisters, especially for newly synthesized proteins. Imagine a protein being born, right? Emerging from the ribosome is this long, floppy, unstructured chain of amino acids.
That's not very functional.
Not at all. To become functional, it needs to fold into a very specific, complex, three-dimensional shape. Molecular chaperones are like these highly skilled architects, or maybe personal trainers.
Guiding the folding process?
Exactly. Guiding these new proteins through that intricate process, ensuring they fold into their correct functional configurations. Like an origami master, carefully guiding paper into a precise form.
Okay, so they help with the initial folding. What else?
Well, their vital role doesn't stop there. Equally critical is their secondary function, preventing inappropriate aggregation.
Aggregation, like clumping.
Exactly. This is a cellular nightmare scenario where proteins, instead of folding correctly, just clump together uselessly. Or even worse, form toxic masses that can seriously damage the cell.
So chaperones are like anti-clumping agents.
That's a good way to think about it. They ensure proteins remain soluble, functional, and, well-behaved. A prime example of these molecular chaperones are the heat shock proteins, or HSPS.
Heat shock proteins, does that mean they only work under heat stress?
That's where their name comes from, because cells produce more of them when exposed to heat. But it's really important to understand they are absolutely critical for everyday protein health, maintaining stability even under normal conditions too.
Got it. So HSPs are key players.
Yeah.
And speaking of stress, you mentioned a specific pathway.
Right. There's a truly fascinating signaling mechanism directly tied to this, the HSF1 pathway, or the heat shock factor 1 pathway.
HSF1.
This pathway is essentially the cell's highly sensitive, incredibly responsive quality control alarm system.
Like a smoke detector for misfolded proteins.
Exactly like that. Normally HSF1, which is the main transcription factor responsible for switching on chaperone genes.
So it controls making more chaperones.
Yes. Normally it's kept inactive. It's often bound to chaperones like HSP90, which essentially keeps HSF1 busy and unable to activate gene expression. Think of HSP90 as a security guard, keeping HSF1 on a short leash.
Okay, so HSF1 is usually held back.
But here's where the alarm system kicks in. When misfolded proteins start to pile up in the cell signaling cellular stress, much like smoke filling a room and overwhelming the available fire extinguishers.
The chaperones are overwhelmed.
Right. HSP90, realizing there's a much bigger problem at hand, releases HSF1.
It lets go of the leash.
Precisely. This liberation allows HSF1 to become active. And once active, HSF1 doesn't waste any time. It travels to the cell's nucleus and induces the expression of more chaperones.
Calling in reinforcements.
Essentially, yes. Calling in massive reinforcements to handle the increased load of misfolded proteins and restore order. It's a rapid, highly responsive, and incredibly intelligent system designed to restore cellular balance and prevent widespread damage.
Okay, that's the chaperone response. But what happens if a protein is just too far gone, severely damaged, beyond repair by chaperones?
Good question. That's where the second major component of the proteostasis network steps in, the ubiquitin proteasome system, or UPS, the selective shredder.
The UPS, not the delivery company, I assume.
No, definitely not. The UPS is the cell's primary degradation system, specifically designed for individual proteins or relatively small protein complexes.
So smaller stuff.
Right. Think of it as a highly sophisticated molecular shredder, but with incredible specificity and precision. It's designed for the rapid, targeted removal of proteins that are no longer needed, are misfolded, or have become damaged beyond rescue.
Like a high-security paper shredder for cellular documents.
That's a perfect analogy, ensuring swift and complete disposal.
How does it work, this tagging and shredding?
It's quite elaborate, actually. A multi-step process. First, the damaged or defective proteins are literally tagged for destruction.
Tagged how?
This tag is a small, incredibly conserved protein called ubiquitin. It's found in almost all tissues of eukaryotes.
Ubiquitin.
Like ubiquitous. Exactly. It's like putting a little bright orange sticky note or a very specific barcode on something that absolutely needs to be thrown out and destroyed. Without this tag, the protein is generally safe.
So the tag is the key signal.
It is. And this tagging process relies on an incredibly sophisticated enzymatic cascade, like a molecular assembly line involving 3 key types of enzymes, each playing a distinct and absolutely crucial role.
3 enzymes, E1, E2, E3.
You got it. You have E1, the ubiquitin activating enzyme. It starts the whole process by activating ubiquitin itself, sort of energizing it and getting it ready.
Step 1, activate the tag.
Then E2, the ubiquitin conjugating enzyme, carries that activated ubiquitin, acting like a shuttle.
Okay, step 2, carry the tag.
But the real precision and intelligence comes from E3, the ubiquitin ligase. This is the truly smart part of the system.
Why is E3 the smart one?
Because it's the enzyme that actually recognizes the specific target protein destined for destruction and transfers the ubiquitin tag, or usually multiple tags, onto it. It's the E3 ligase that provides the critical specificity.
Ah, so E3 makes sure only the right proteins get tagged.
Precisely. It ensures only the correct, problematic proteins are marked for removal, preventing, indiscriminate cellular destruction. There are hundreds of different E3s, each recognizing different signals on proteins.
Wow, okay, so the protein is tagged by E1, E2, and specifically E3. Then what?
Once tagged, these ubiquitinated proteins aren't just left floating around. They are precisely delivered to a remarkable molecular machine called the 26S proteasome.
The proteasome, that's the shredder part.
That's the shredder. It's a large barrel-shaped molecular complex, a multi-subunit proteolytic machine. Once the tag protein gets inside,
gets shredded.
Essentially, yes. It functions as a powerful molecular shredder, meticulously breaking down the ubiquitinated proteins into small peptides. These peptides can then be further recycled back into individual amino acids, ready to be reused for synthesizing new healthy proteins.
A closed-loop system. Very efficient.
Extremely. The continuous and efficient operation of the UPS is absolutely critical for preventing the accumulation of defective proteins. Without it operating flawlessly, these proteins would rapidly build up, leading to toxic aggregates and widespread cellular dysfunction, like a garbage disposal backing up.
Right, causing a huge mess.
Exactly. However, and this is a crucial distinction, it really highlights the cell's multi-layered strategy. The UPS has inherent size limits.
Size limits, meaning it can't handle everything.
Right. It excels at individual proteins or small complexes, but it literally can't handle massive clumps of proteins, large insoluble aggregates, or entire damaged organelles. They just won't fit.
So if the UPS is for small trash, what handles the big stuff?
That's precisely where our next complementary system comes in.
Which brings us to the autophagy lysosome system, the cellular recycling plant, and large waste disposal.
Exactly. You just explained that the UPS is great for small, individual pieces of trash. So when the UPS reaches its capacity, when it's simply unable to handle those large protein aggregates or entire defective organelles that are too big to fit into its shredder,
autophagy steps in.
The autophagy lysosome system steps in. This is the cell's designated mechanism for clearing larger debris. You could think of it as a cellular landfill or large waste disposal unit.
But you also mentioned recycling.
Yes, and that's critical. It also has a significant and crucial recycling capacity. It's about breaking down large components into their basic building blocks for reuse by the cell.
So not just disposal, but reclamation.
Precisely. Autophagy, which literally translates from Greek as self-eating, is a fundamental and ancient process, absolutely essential for cellular maintenance, survival, and adaptation. It's a key strategy cells use to survive periods of stress and to continuously renew themselves.
And there are different types.
There are 3 main types, each with unique mechanisms, but they all work together to ensure comprehensive cellular hygiene.
Okay, what's the first type?
First, and probably the most widely recognized, is macroautophagy. In common conversation, this is often just called autophagy.
Macroautophagy, the main one.
It's the most common and prominent form. It involves specific portions of the cytoplasm, including those problematic protein aggregates or even entire defective organelles like a worn-out mitochondrion.
Being captured somehow.
Yes, being encapsulated by a novel double membrane vesicle that forms around them. We call this vesicle an autophagosome.
Autophagosome, the double-layered bag.
Kinda, yeah. This autophagosome then travels through the cell, specifically recognizes and fuses with a lysosome.
The lysosome, that's the stomach of the cell, right?
Essentially, yes. It contains a formidable array of powerful enzymatic breakdown machinery. So the autophagosome delivers its cargo to the lysosome for destruction. It's like a specialized recycling truck, picking up a big bag of mixed waste and delivering it to a processing plant for complete breakdown and reclamation.
And this macroautophagy, is it always picking up specific things?
Well, it demonstrates remarkable versatility. It can operate in both non-selective and highly selective modes.
Non-selective, like indiscriminate.
Exactly. In times of nutrient deprivation or starvation, when the cell desperately needs energy, cells often employ non-selective macroautophagy.
So it just eats whatever's nearby.
Pretty much. During this phase, they non-selectively engulf and recycle random cytoplasmic components, including healthy proteins and organelles, just to get fuel. It acts essentially as an internal fuel source, allowing the cell to survive periods of scarcity. It's a critical, though maybe drastic, survival mechanism.
Okay, that's for starvation. What about normal conditions?
Under normal conditions, and especially during cellular stress, macroautophagy becomes incredibly targeted and precise. This is called selective macroautophagy.
Selective, like it has specific targets.
Exactly. For example, agriphagy is the targeted removal of harmful protein aggregates.
Ah, like the ones in neurodegenerative diseases.
Precisely. Those sticky, toxic clumps, like the tau aggregates in Alzheimer's or the alpha-synuclein aggregates in Parkinson's. Agriphagy is the cell's specialized defense against these molecular tangles.
What other specific targets are there?
Beyond protein aggregates, autophagy has other highly specific targets. There's mitophagy.
Mitophagy, eating mitochondria.
Exactly. The specific clearance of old, damaged, or dysfunctional mitochondria. Now, we won't dive deep into mitochondrial health today, but it's a critical example of autophagy's selective nature in maintaining organelle quality control.
Makes sense. Keep the powerhouses running smoothly.
And critically, there's xenophagy.
Xenophagy. Xeno meaning foreign.
Yes. This is the vital process of clearing intracellular pathogens, like invading bacteria or viruses. It's a key part of the innate immune response, showcasing autophagy's essential role in cellular defense and eliminating invaders.
Wow, so it's waste disposal, recycling, energy generation, and even defense. Pretty versatile.
Incredibly versatile. That was macroautophagy. Then we have microautophagy.
Micro, smaller scale.
Yes, and it operates differently. This is a more direct process where the lysosome membrane itself simply invaginates, meaning it folds inward, to directly engulf small bits of cytosol, the liquid part of the cytoplasm.
So the lysosome itself takes a bite.
Kind of, yeah. It's like the lysosome is taking tiny direct bites rather than waiting for a separate vesicle like the autophagosome to form. It's less about large cargo and more about continuous sampling and recycling of general cytoplasmic content.
Okay, direct nibbling. And the third type?
And finally, there's chaperone-mediated autophagy, CMA.
CMA, chaperone-mediated. So chaperones are involved again.
Yes, but in a very different way. This is a highly selective pathway, quite distinct in its mechanism from macroautophagy. Its elegant mechanism involves specific proteins, and only those proteins that possess a particular KFEQ-like motif in their amino acid sequence.
KF or Q, that's specific, like a barcode.
Exactly. Think of this motif as a specific molecular barcode or zip code that signals them as targets just for this pathway. These tagged proteins are recognized by a specific chaperone, HSC70.
Okay, HSC70 finds the barcode. Then what?
This chaperone then, in a remarkable act of precision, threads these specific proteins directly into lysosomes through a specialized receptor protein on the lysosomal membrane called LMP2A.
Threads them through, like pulling thread through a needle.
That's a good analogy. It's a very precise protein-by-protein degradation system, often used for degrading specific soluble, long-lived proteins when needed.
Wow. Okay, so 3 types. Macro for big stuff, selective or not. Micro for small sips. And CMA for specific barcoded proteins.
You've got it. And what's truly incredible is how all 3 autophagy pathways, macroautophagy, microautophagy, and CMA coexist and work together in most cells.
A coordinated team.
Exactly. Ensuring continuous cellular self-cleaning and adaptation. It's truly a multi-layered, redundant system, providing multiple lines of defense. And under various stress conditions, whether it's heat oxidative stress, or nutrient deprivation, cells dramatically ramp up these pathways.
We ramp them up.
To efficiently dispose of damaged components and recycle materials vital for survival and cellular resilience, it's a key stress response.
And how do autophagy and the UPS, the shredder, work together? You said they were complementary.
What's absolutely crucial to understand is the profound synergy between them. They're not competing systems. They are highly collaborative and indispensable partners.
So UPS handles the small stuff.
Right, the trash that is small, soluble, and individual, like single misfolded proteins. But when you have bigger garbage, like large, insoluble protein aggregates that might have formed despite chaperone efforts or entire defective organelles.
That's when autophagy takes over.
Exactly. That's precisely where autophagy steps in, efficiently hauling them to the lysosomes for bulk degradation and recycling.
Is there a direct link? Like a handoff mechanism?
There is. There are even fascinating molecular bridges that physically connect these two seemingly distinct systems. A superb example is an adaptoprotein called P62SQSTM1.
P62, what does it do?
This protein actually acts as a literal molecular bridge. It binds directly to ubiquitin-related protein aggregates. Remember the ones tagged by the UPS?
But we're too big for the UPS shredder.
Exactly. P62 binds to those tag aggregates and then helps to funnel them directly into nascent autophagosomes.
So it connects the UPS tag to the autophagy machinery?
Precisely. This critical handoff ensures that even the large, complex aggregates are efficiently recognized and cleared by autophagy after the UPS has done its initial work of tagging. It's like one cleanup crew flagging an oversized item for the next specialized crew to pick up.
Okay, let's take a breath here. That was a lot of detail. Let's try and unpack this. We've just navigated the incredibly intricate world of cellular quality control, from molecular chaperones, guiding new proteins, acting as first responders.
The architects.
Right. To the specialized shredding capability of the UPS for individual proteins.
The selective shredder.
And then to the large-scale cleanup and recycling operations of the autophagy lysosome system for the bigger debris.
The heavy-duty cleanup and recycling plant.
Essentially, we've seen in meticulous detail how cells are constantly, dynamically, and intelligently working to keep themselves pristine and functional. It's pretty amazing.
What's truly fascinating here, if we step back for a second, is understanding that these aren't isolated processes just operating in silos. They are an intricately interconnected network, the proteostasis network.
Right, the PM.
Constantly adapting, communicating, responding to the cell's immediate needs and various environmental stresses. The critical takeaway, the really profound insight here, is that their combined, coordinated efficiency dictates whether a cell stays healthy, vibrant, and functional, or, conversely, starts to accumulate problematic waste, becoming a dysfunctional mess that eventually breaks down.
Which brings us to the critical dichotomy. Cleaning up versus clogging up. It sounds like the ultimate binary consequence of how well these systems perform.
It really is. It determines cellular destiny and ultimately, you could argue, our own health trajectory.
How so? Let's start with the positive side.
Okay. If a cell maintains a robust, efficient proteostasis network, it ensures cells remain cleaned up.
Cleaned up meaning?
Meaning proteins are correctly folded, damaged components are swiftly removed, and everything within the cell functions optimally and fluidly. It's a clear picture of cellular health, resilience, and vitality, like a well-maintained high-performance machine.
And the flip side, if the network fails.
Conversely, if this intricate network begins to fail, it leads directly and pretty much inexorably to clogging up.
Clogging up, like a drain.
Exactly. This manifests as dangerous pathological accumulations of misfolded proteins and cellular junk. These accumulations directly impair cellular function, creating widespread dysfunction, acting like a thick, impeding sludge, eventually affecting entire tissues and organs, leading to a cascade of problems.
Can we see this play out in real life?
Oh, absolutely. We see this dramatic contrast play out vividly in real-world scenarios, starkly contrasting the devastation of disease with the remarkable resilience of exceptional longevity.
Give us an example of failure.
Many devastating neurodegenerative diseases like Alzheimer's, Parkinson's, and Huntington's are archetypal, tragic examples of this cellular cleaning failure.
How so?
In these conditions, toxic protein aggregates accumulate relentlessly within the highly sensitive neurons. Why? Because the proteostasis systems are either overwhelmed by the sheer volume of misfolded proteins or are themselves impaired and failing.
So the cleanup crew is either swamped or broken.
Exactly. This direct accumulation of misfolded proteins leads directly to profound neuronal dysfunction and ultimately neuronal death, resulting in the debilitating and irreversible symptoms of these diseases. It's a clear consequence of cellular garbage overwhelming the system.
That's a grim picture. What about the success story, the flip side?
But the flip side of this coin is just as compelling and maybe even more hopeful. Contrast this grim picture with the remarkable observations in individuals who age exceptionally healthily, particularly some centenarians.
People living healthily past 100.
Right. It's not just that they live longer. They appear to maintain significantly more youthful proteostasis activity. There's a specific compelling study that found fibroblast cells.
Skin cells, basically.
Well, connective tissue cells found throughout the body. Fibroblasts from healthy centenarians retained proteasome subunit levels and activity akin to young cells.
Like those of young people.
Exactly. This is distinctly unlike cells from typical older adults, who universally show a noticeable decline in these critical cleanup systems.
So centenarians keep their cellular cleanup crews working like they're young.
This powerful finding strongly suggests that their protein clearance systems remained closer to a young equilibrium, a state of youthful, robust function. And it's hypothesized this contributes significantly to their exceptional longevity and their remarkable ability to resist age-related diseases.
So there's a clear link to lifespan.
Undeniable. Enhanced proteasome and autophagy activity is robustly correlated with extended lifespan across multiple organisms, from simple yeasts to complex mammals. Conversely, a decline in these essential clearance mechanisms is directly associated with accelerated aging and a higher incidence of age-related diseases. It's a powerful, fundamental testament to the profound importance of efficient cellular cleanup for overall health and lifespan.
Okay, so we've really built a foundational understanding here of the proteostasis network. We appreciate how molecular chaperones, the UPS, and the diverse forms of autophagy work in this intricate concert as the cell's cleanup crew.
A coordinated effort.
This sophisticated, interconnected network ultimately determines whether our cells remain pristine and functional or become overwhelmed by molecular junk.
The clogging up scenario.
Right. And the profound implications are crystal clear. Effective cellular cleaning is not just about tidiness or efficiency. It's a cornerstone of healthy aging and a powerful bulwark against devastating diseases like neurodegeneration. It's about maintaining the integrity of life itself, right down at its most fundamental level.
Absolutely. And maybe this is a good point for one of those flashcard recaps to consolidate all that rich information.
Great idea. Let's run through the key takeaways from this first big section. Think of these as the fundamental principles we've just uncovered. You start.
Okay. First, proteostasis. It's about maintaining cellular protein balance in their correct shape, location, and precise amounts.
Got it. Balance. Next.
The proteostasis network, PN, itself. This comprehensive system includes molecular chaperones, the ubiquitin proteasome system, UPS, and the autophagy lysosome system, the whole toolkit.
Chaperones, UPS, autophagy. Okay. What about chaperones specifically?
Chaperones, like HSPS. These molecular guides assist in correct protein folding and crucially prevent harmful protein aggregation. Their activity is dynamically activated by the HSF1 pathway during cellular stress.
Guides and protectors activated by stress, then the UPS.
Ubiquitin proteasome system, UPS. This system selectively targets individual ubiquitinated proteins for degradation via the 26S proteasome. It's the cell's precise shredder for small, soluble protein trash.
Small trash shredder? Okay. Autophagy.
Autophagy lysosome system. This is the cell's sophisticated recycling plant designed for clearing larger aggregates, entire damaged organelles, and for general cellular renewal. Handles the big stuff.
Big stuff recycler. And the types of autophagy.
Right. Macroautophagy. The most common form using autophagosomes plus lysosomes can be non-selective, starvation, or highly selective, agraphagy, mitophagy, xenophagy.
Then microautophagy. That's the direct lysosome invagination, the nibbling.
In CMA, chaperone-mediated autophagy, highly selective HSC70 chaperone-mediated threading of specific proteins with that KFERQ-like motif into lysosomes via LAMP2A, the barcode system.
Okay. And how do UPS and autophagy work together?
UPS and autophagy complementarity, they're synergistic. UPS handles small, soluble waste. Autophagy manages large, aggregated, or organellar waste. Adapter proteins like P62, SQSTM1 act as crucial bridges between them.
The handoff. What happens when it fails?
Failing PN, clogging up. This leads directly to the accumulation of toxic protein aggregates, a hallmark pathology in neurodegenerative diseases like Alzheimer's, Parkinson's, Huntington's.
And when it works well?
Robust PN cleaned up A well-functioning proteostasis network contributes significantly to healthy aging and exceptional longevity, like in those healthy centenarians maintaining youthful proteasome activity.
And the bottom line correlation.
Crucial correlation. Enhanced cellular clearance capacity is strongly correlated with extended lifespan, while the decline in these systems directly accelerates aging.
Perfect summary. Now let's pivot and delve even deeper into one specific aspect of this incredible machinery: autophagy as a hallmark of aging. Cleaning up declines with age. This is where I think the plot thickens considerably.
Oh, definitely. It reveals the profound implications for our own health as we age.
What's the significance here? Why is it called a hallmark?
The significance here cannot be overstated, really. It's a concept that has truly transformed our understanding of aging. The loss of proteostasis and disabled macroautophagy are not just observed phenomena that happen to coincide with old age.
They're not just symptoms.
No. They are formally recognized as hallmarks of aging by leading cellular biologists and gerontologists. This designation isn't given lightly.
What does it signify?
It signifies their universal observance in old age across diverse species, from simple yeast to complex mammals, and critically, their strong causal link to aging.
Causal link, meaning they cause aging.
Meaning accelerating their dysfunction, demonstrably shortens lifespan across species, while enhancing them extends it. This isn't just a symptom, it's considered a fundamental driver of the aging process itself.
Wow. Okay. And you mentioned it declines with age.
Yes, and the really insidious, almost paradoxical part is the age-related decline of cellular housekeeping. It's not usually a sudden, catastrophic collapse. It's more an insidious, gradual yet relentless loss of control and efficiency as organisms age.
How does this decline manifest? What do we actually see?
We observe very specific, measurable manifestations. Chaperone levels, those protein architects, often decrease. The inherent deficiency of proteasomes, our molecular shredders, and lysosomes, our recycling plants, demonstrably wanes. And perhaps most critically, overall autophagy activity, that broad self-eating process, measurably slows down and becomes less effective.
It's like the entire cleanup crew starts to fatigue and lose coordination over time.
That's a great way to put it. And this age-related decline creates a truly perilous feedback loop, a vicious cycle. This is one of those profound insights into cellular aging.
A vicious cycle? How does that work?
As damaged proteins and organelles accumulate because the clearance systems are impaired, they don't just sit there passively waiting for disposal. They, in turn, actively gum up or directly inhibit the very systems, the chaperones, the proteasomes, the lysosomes, the autophagy pathways that are desperately needed to clear them.
So the garbage breaks the garbage disposal.
Exactly. It's like a perpetually jammed garbage disposal unit where the garbage itself actively breaks the mechanism designed to remove it. This creates a self-perpetuating cycle of cellular dysfunction and increasing damage. It's a core mechanism of biological aging.
Is there direct evidence for this, for autophagy getting stalled?
Absolutely. We have compelling, concrete evidence of stalled autophagic flux observed consistently in aging cells and tissues.
Stalled autophagic flux, what does that mean exactly?
It's a technical term, but it vividly describes a cellular cleanup process that is severely congested and incomplete. Researchers consistently observe a significant buildup of P62.
P62, the bridge protein.
Right, and a backlog of undegraded autophagic cargo. If P62 builds up in the cell, it's a clear, unequivocal sign that the autophagosomes aren't properly fusing with lysosomes, or that the lysosomal degradation itself isn't happening efficiently.
The trucks are full, but the recycling plant is backed up.
Precisely. Additionally, in aging rodent tissues and various cellular models, researchers consistently observe a marked, reduced expression of core autophagy genes.
Genes needed to make the autophagy machinery.
Exactly. These aren't just any genes. They are absolutely crucial for initiating, forming, and executing the entire autophagy process. We're talking about genes such as ATG5, ATG7, and Beclin-1, along with key proteins like LC3 and ULK1.
All essential components.
Absolutely essential for autophagosome formation and function. Without adequate levels, the whole process effectively grinds to a halt. And furthermore, as if that weren't enough, chaperone activity and the crucial heat shock response mediated by HSF1 also decline with age.
Making the initial problem of misfolded proteins even worse.
Right, exacerbating the problem by allowing even more misfolded proteins to accumulate unchecked.
So what's the overall consequence of all this decline and stalling?
The vivid consequence is an overloaded cellular garbage disposal. Cells literally end up with a pathological accumulation of substances like lipofuscin, often called age pigment.
Lipofuscin, what is that?
It's essentially indigestible waste products that build up stubbornly within lysosomes, kind of like cellular rust. You also see massive problematic protein aggregates, those toxic clumps, and an ever-increasing number of dysfunctional mitochondria.
Which are not only inefficient, but potentially harmful.
Right, they can produce harmful reactive oxygen species. All of these accumulated unresolved components are significant direct contributors to age-related pathology and disease. They drive everything from neurodegeneration to metabolic disorders and overall organ failure. It's a systemic breakdown.
You mentioned earlier this isn't just correlation, but causation. How do we know the decline causes aging?
Now, what's truly powerful about this age-related decline in cellular cleaning machinery is that it is not merely correlative. It's not just something we observe happening at the same time as aging. Robust, meticulous, experimental evidence across a variety of species definitively shows it is causal.
It directly causes aspects of aging.
Yes, this is where scientific inquiry moves from observation to direct intervention and proof.
How can we prove it? Through intervention.
Exactly. We can prove this through direct intervention, demonstrating a profound cause and effect relationship. For instance, partial inhibition of proteasomes or autophagy in adult animals often dramatically shortens lifespan and induces premature aging phenotypes.
Wow. So deliberately crippling these systems makes animals age faster.
It's a powerful, undeniable finding. If you deliberately cripple these cellular cleaning systems, even partially, the organisms age faster, become frail, and die younger. It's a stark demonstration of their fundamental importance.
Are there specific examples?
Oh, yes. Specific, compelling examples across diverse modern organisms. In C. elegans, worms or fruit flies, which are common workhorses in aging research.
Easy to study, right?
Right. Researchers have meticulously shown that knocking down autophagy genes, meaning genetically reducing their expression or disabling them, genes like ATG1, ATG7, etc., directly, unequivocally shortens their lifespan. They literally do not live as long.
And in mammals, like mice.
Yes, in mice, those that are engineered to have reduced autophagy, for example, by knocking out the ATG5 gene in certain tissues, consistently exhibit accelerated tissue aging and functional decline. The evidence is really overwhelming across species.
Okay, so crippling the system shortens life. What about the opposite?
But here's the truly optimistic and transformative counterpoint, the flip side of that coin. Boosting proteostasis extends longevity.
So if crippling shortens life, boosting extends it.
Exactly. If crippling the system dramatically shortens life, then conversely, genetically or pharmacologically boosting it can reliably extend it. This has been shown repeatedly through direct genetic enhancement experiments.
Like what?
Worms or flies live measurably longer when proteasome subunits are overexpressed, meaning they have more of those molecular shredders, or when autophagy is genetically enhanced, making their cellular cleaning processes more efficient and robust.
Okay, genetic boosts work. What about drugs? Can we boost it pharmacologically?
And even more exciting, perhaps from a therapeutic perspective, are the pharmacological boosters with proven geroprotective effects that have been identified and are currently under intense study.
Geroprotective meaning anti-aging.
Essentially, yes, protecting against aging. One of the most significant and well-studied examples is rapamycin.
Rapamycin, I've heard of that.
It's a powerful mTOR inhibitor. Remember mTOR, the nutrient sensor.
Right, involved in controlling autophagy initiation.
Exactly. By inhibiting mTOR, rapamycin reliably extends lifespan in mice. And it does so, at least in large part, by significantly upregulating autophagy.
So turning up the recycling.
Yes. Now, rapamycin does have other complex effects on various cellular pathways and immune function, but the activation of autophagy is considered a major and well-established contributor to its remarkable anti-aging properties. Are there other compounds? Another powerful and widely studied example is spermidine. It's a naturally occurring polyamine compound. It has been consistently shown to extend lifespan across a broad spectrum of organisms, including yeast, worms, flies, and mice.
Wow, across many different species. How does it work?
And critically, it achieves these longevity benefits primarily, though not exclusively, via its ability to activate autophagy. It's a remarkable and consistently observed effect across such diverse species, suggesting a fundamental conserved mechanism is at play.
So genetic boosts, drug boosts. What about naturally long-lived creatures?
These insights are further solidified by observations from naturally long-lived individuals and species. It appears that long-lived individuals, like the centenarians we discussed,
With the youthful proteasomes.
right, And exceptionally long-lived species in the animal kingdom seemingly maintain their cellular cleanup crew running more efficiently, more youthfully for much longer. We already mentioned the centenarian fibroblasts.
Any animal examples?
A truly fascinating example, often studied in aging research, is the naked mole rat.
Yes, the weird-looking, long-lived rodents.
Exactly. They are exceptionally long-lived rodents. They can live for over 30 years, which is astonishing for a rodent of their size. Far exceeds what you'd expect.
And their cleanup crews?
And notably, they consistently show higher proteasome activity and autophagy levels compared to shorter-lived mice, their common lab counterparts. This strongly suggests a biological advantage in cellular maintenance and waste disposal contributes significantly to their remarkable longevity and resistance to many age-related diseases.
Okay, so boosting autophagy seems key. Does it relate to other known longevity strategies?
That leads to another critical connection, drawing parallels between different longevity pathways. Autophagy upregulation mimics longevity pathways.
Meaning activating autophagy has similar effects to things like calorie restriction.
Precisely. Genetic manipulations that upregulate autophagy can effectively mimic the profound lifespan extension effects of caloric restriction or reduced insulin signaling. These are both well-established, classic longevity pathways studied for decades.
So autophagy might be how those pathways work, at least partly.
It suggests that autophagy is not just a mechanism, but potentially a convergent mechanism. It's one of the key fundamental ways these other successful longevity strategies exert their magic, by enhancing cellular cleaning and renewal.
Do we have specific genetic proof of this mimicry?
Yes. For specific genetic examples, mice that were overexpressing at G5, remember, a crucial autophagy protein,
Right.
demonstrated increased autophagy and consistently lived longer than controls. This wasn't just correlation, it was direct causal proof through genetic engineering.
And in worms.
In worms, overexpressing HLH30, which is the worm equivalent of a human protein called TFE. TFE is a master regulator of lysosomal and autophagy genes. Overexpressing its worm equivalent, HLH30, also extended lifespan. This evidence from genetic manipulation provides powerful proof of concept.
Okay, wow. Here's where it gets really interesting and actually quite profound, doesn't it? It's not simply that cellular junk accumulates as we age.
No, it's more complex than that.
The very systems designed by evolution to clean it up decline in their efficiency with age. We're talking about that vicious, self-perpetuating cycle where the garbage literally starts to clog the disposal unit.
Exactly. It's a downward spiral.
But the powerful, hopeful implication here is that by understanding these mechanisms and perhaps even finding ways to boost or maintain these cleaning systems, we're seeing profound, measurable effects on lifespan and health across the entire animal kingdom. It suggests a universal principle.
Right. And if we connect this to the bigger picture, the formal recognition of disabled proteostasis and macroautophagy as official hallmarks of aging is critical. It moves us definitively from mere observation of age-related phenomena to a deep understanding of direct causation.
From correlation to cause.
Yes. The extensive experimental evidence, from the simplest worms to complex mice, clearly, unequivocally demonstrates that maintaining or enhancing this cellular cleanup capability is a strong, indeed, a fundamental determinant of how well and how long an organism lives.
Which raises a huge question.
It raises an incredibly important question, not just for science, but for all of us. What does this powerful insight mean for potential interventions that could target these processes in humans, aiming for a longer, healthier life?
The ultimate take-home message, then, the central principle distilled from all this evidence, seems incredibly profound, maybe one of the most significant insights in aging research.
I think so. It's that clearance capacity is a strong, indeed, an overriding determinant to aging rate.
Clearance capacity determines aging rate.
Precisely. If cells can continue cleaning up effectively, maintaining that pristine, highly organized internal environment, then tissues and organs throughout the body simply stay functional longer.
Which contributes not just to lifespan, but health span.
Exactly. It contributes not just to an extended lifespan, but crucially, to what we define as health span, the period of life spent in good health, free from chronic disease and disability. That's arguably the more important goal.
And the alternative.
Conversely, if cells clog up, if their intricate garbage disposal and recycling systems falter and fail, the inevitable result is widespread cellular dysfunction, the accumulation of toxic waste, and the earlier onset of age-related disease. It's a fundamental biological principle with immense direct implications for our health as we age.
And a powerful target for future therapy.
The very powerful target.
Okay, so we've now rigorously seen that the decline of cellular cleanup is not just a mere symptom, but a core causal driver of aging, recognized as a formal hallmark.
Right.
The accumulating evidence, both observational and experimental, clearly links a faltering proteostasis network and disabled autophagy to shortened lifespans and the progression of age-related pathologies.
The downside is clear.
But crucially, and this is where the hope lies, we've also discovered that robustly boosting these internal cleaning systems, whether through precise genetic enhancement or strategic pharmacological activation, can reliably extend longevity across diverse species.
The potential upside.
Which offers a profound, actionable take-home message. The cell's inherent ability to clean house effectively is a powerful, perhaps even the most powerful, predictor of its and our own aging rate.
Well summarized. Ready for the flashcard list for this chapter?
Let's do it. Consolidate these critical insights about aging and maintenance. You start again.
Okay. Key point 1. Loss of proteostasis and disabled macroautophagy. These are not just observations. They are officially recognized hallmarks of aging, indicating their causal role.
Hallmarks. Got it. What happens with age?
Age-related decline. Aging predictably causes a gradual decline in the efficiency and levels of chaperones, proteasomes, lysosomes, and overall autophagy activity. Everything slows down.
The cleanup crew gets tired.
Ugh.
And the vicious cycle.
Vicious cycle. A key insight: damaged components accumulate due to impaired clearance. And this accumulation in turn further gums up and inhibits the very cellular cleaning systems. A self-perpetuating decline.
How do we see this gumming up?
Evidence of stalled autophagy. Observable markers like the persistent buildup of P62, accumulation of undegraded autophagic cargo, and reduced expression of core at G genes, like at G5, at G7, Becklin-1, LC3, ULK1, all indicate a congested and incomplete cleanup.
The system is backed up. Consequences.
Consequences of overload. Results in an overloaded cellular garbage disposal, leading to pathological accumulation of levofuscin, age pigment, massive protein aggregates, and dysfunctional mitochondria, all contributing to age-related pathology.
And the proof it's causal.
Causal evidence. Direct experimental proof shows that even partial inhibition of proteasomes or autophagy in adult animals consistently shortens lifespan and induces premature aging phenotypes. For example, in C. elegans, fruit flies, at G5 knockout mice.
What about boosting it?
Boosting proteostasis extends longevity. The optimistic counterpoint. Genetic enhancement, overexpressing proteasome subunits or autophagy genes, and pharmacological activators like rapamycin spermidine demonstrably extend lifespan across multiple species.
And long-lived creatures.
Insights from long-lived organisms. Exceptionally long-lived species like naked mole rats and healthy human centenarians show demonstrably higher or more youthful activity in these vital cellular maintenance systems.
How does it relate to things like calorie restriction?
Autophagy upregulation mimics longevity pathways. Critically, genetic manipulations that specifically upregulate autophagy effectively mimic the lifespan extension seen in classic longevity pathways like caloric restriction-reduced insulin signaling, for example, at G5 overexpression mice, HLH30TFEB in worms.
And the absolute core principle.
Core principle. The cell's innate clearance capacity is a strong and fundamental determinant of the aging rate. When cells are cleaned up, they remain functional longer. When they clog up, dysfunction and disease inevitably follow.
Excellent. That really captures the essence of why this matters so much for aging. Now let's really zoom in on one of the central players we've discussed today. How macroautophagy works self-eating in action.
Okay, getting into the nuts and bolts.
Right. This process is so vital, so fundamental to cellular health, that it's formally deemed a hallmark that goes awry with aging. So it truly deserves a closer, detailed look at its intricate molecular mechanics. We're going inside the machine itself.
Absolutely. Macroautophagy, despite its seemingly simple outcome of cellular recycling, involves several highly coordinated, precise, and actually quite energetically demanding steps within the cell. It's a real marvel of molecular choreography.
Let's start right at the beginning with 1. Initiation, the phagophores' first glimmer.
The very start.
This is the very first, almost miraculous step in this complex process. Imagine, if you will, a tiny, flexible, cup-shaped membrane, almost like a crescent moon, beginning to spontaneously form inside the cell's cytoplasm.
Out of seemingly nowhere.
Well, from lipid sources in the cell. This is what we call an isolation membrane, or a phagophore, and it doesn't form randomly. It actively begins to precisely wrap itself around a specific portion of the cytoplasm.
The stuff it needs to get rid of.
Exactly. Containing the material destined for degradation, whether it's a damaged protein aggregate, a worn-out organelle, or even an invading pathogen, it's actively reaching out to engulf its target.
What kicks this off? What's the trigger?
The kickoff switch for this entire intricate process, the molecular initiator that orchestrates this membrane formation, is a critical player known as the ULK1-AG1 kinase complex.
ULK1 in mammals, AG1 in yeast, highly conserved.
Right. ULK1 is the mammalian equivalent of the AG1 gene found in yeast, which was one of the very first autophagy-related genes discovered. Highlights how ancient this pathway is.
So this complex acts as the master switch.
Exactly. Orchestrating the initial steps of phagophore formation, signaling the cell to begin the engulfment process. And what's truly fascinating, and a testament to the cell's remarkable intelligence, is the sophisticated control.
Control by
Nutrient sensors tightly control this step.
Bringing us back to MTOR and AMPK.
That's right. This is where cellular intelligence truly shines. Briefly, those two major pathways, MTOR and AMPK, act as key nutrient and energy sensors. They effectively tell the cell whether it's in a fed state or a state of deprivation.
Rich or poor, energy-wise.
Pretty much. Depending on the cell's precise energy status, these sensors exert exquisite influence over ULK1A and AG1 activity, directly determining whether autophagy is initiated.
So if the cell is starved,
if nutrients are scarce and energy reserves are low, MTOR activity goes down and AMPK activity goes up. Both of those changes effectively activate ULK1.
signaling the cell to ramp up recycling.
Exactly, to generate internal fuel and clear debris. It's an exquisite example of a cell precisely, dynamically responding to its environment, making intelligent resource allocation decisions.
So as you visualize it, picture that tiny, flexible crescent moon, the phagophore, beginning to form inside the cell, poised to embrace its specific cellular components. Its formation isn't random.
No, highly controlled.
It's a precise molecular dance, signaled by master complexes like ULK1, and remarkably, it's exquisitely sensitive to the cell's moment-by-moment energy levels. A beautifully orchestrated and highly intelligent beginning to the cellular recycling process.
Okay, so that's initiation. The membrane starts forming. Where does it go from there? According to the outline, the next detailed step is closure.
Right, skipping ahead slightly in the full biological process, perhaps for brevity, but focusing on the key outline steps. From that crucial initiation, we move to 4. Closure, sealing the autophagosome.
Sealing the deal.
This is the culmination of the autophagosome formation process, the critical moment where the bag is completely sealed. That crescent moon-shaped phagophore doesn't just embrace its cargo, its edges of the membrane meticulously fuse together.
They meet and join up.
Yes. This action seamlessly, perfectly, and robustly seals off a double-membraned autophagosome with its cargo now fully enclosed inside. It's like precisely zipping up a specialized, double-layered, perfectly contained molecular bag, making sure nothing leaks out or in before delivery.
And this completed bag, the autophagosome, does it look different? Can we see it?
It has a truly distinctive visible marker, which is incredibly useful for researchers. From the outside, a completed autophagosome is often visible under a microscope as a spherical vesicle studded with LC32.
LC32, what's that?
LC32 is a lipidated form of a protein called microtubule-associated protein 1 light chain 3. It becomes tightly associated with the autophagosomal membrane during its formation.
So it's a marker that shows up on the autophagosome.
Exactly. The presence of this specific modified form of LC3 on the autophagosome membrane is widely used as a reliable, indispensable marker for active autophagosomes. It provides a clear and direct indication that autophagy is occurring, and these essential cellular recycling units are being formed and matured.
So if you picture that crescent moon we imagined earlier, slowly, deliberately extending its arms,
Extending and curving.
drawing closer and closer until its tips finally meet and seamlessly fuse, creating a perfectly sealed, robust, spherical bubble.
The autophagosome.
This resilient, double-layered vesicle, often recognizable by telltale markers like LC32 decorating its surface, now holds its cargo securely, perfectly packaged, and ready for delivery to the lysosome for complete breakdown and recycling. It's truly a marvel of cellular engineering, a miniature biological machine performing an essential task.
Absolutely.
So if we dive into just these nuts and bolts of macroautophagy itself, initiation and closure, it's clearly a remarkably choreographed and incredibly precise process. We've just walked through its initiation, where that special membrane forms, almost like a cellular mouth, and then how it perfectly seals off its cargo into a fully formed vesicle. It's a testament to the cell's unbelievably sophisticated internal logistics and its ability to construct complex structures on demand.
What's critical to grasp here is not just the individual steps, but the sheer precision at every single stage.
Sure.
From the initial signal from the ULK1 complex, which is tightly regulated by nutrient availability and energy status,
That intelligent control.
to the subsequent growth of that membrane, and then to the final perfect closure of the autophagosome, every single step is vital for the proper, efficient function of this cellular cleanup system. And that LC32 marker gives us a crucial window into observing this dynamic process in action, confirming the formation of these essential cellular recycling units are indeed being made properly.
Okay, another good point for a recap. We've now meticulously traced the intricate journey of macroautophagy, at least its start and finish.
From its precise initiation by the ULK1 anti-G1 complex, a process profoundly influenced by the cell's moment-to-moment nutrient status,
Right.
to the critical, precise closure of the double-membrane autophagosome, which is then perfectly sealed off and identifiable by its unique molecular markers like LC32.
This fundamental self-eating process, we've learned, is not just a cornerstone of cellular health. It is a truly vital player in the entire aging process, directly influencing our longevity and disease susceptibility.
Understanding these detailed steps provides an invaluable blueprint for how cells actively and intelligently manage their internal environment, how they meticulously decide what to preserve and what to recycle for their survival and sustained optimal function.
All right, let's get the flashcard list for this section on macroautophagy mechanics. Your turn.
Okay, macroautophagy, central, essential player in cleanup, and a formally recognized hallmark of aging.
You got it. First step.
Initiation. The process begins with the spontaneous yet precise formation of a cup-shaped isolation membrane, the phagophore, which starts to engulf target cargo.
Kicked off by...
Key initiator.
Yeah.
The ULK1HG1 kinase complex acts as the molecular kickoff switch, orchestrating these initial steps.
And how is it controlled?
Regulation. This initiation step is tightly and exquisitely controlled by crucial nutrient sensors, primarily mTOR and AMPK, influencing ULK1HG1 activity based on the cell's energy status.
Then the final step we covered.
Closure. The phagophore membrane's edges meticulously fuse, sealing off a robust double-membrane autophagosome with its cargo fully enclosed.
And how do we know it's there?
Autophagosome marker. These completed autophagosomes are often visually identifiable as spherical vesicles, studded with the protein LC32, a reliable indicator of active autophagy.
Perfect. Clear and concise. As we wrap up this deep dive, then, let's take a moment to reflect on the truly profound impact of what we've explored today.
Stepping back again.
Yeah. We started by imagining our own cluttered spaces, scaling that concept down to the microscopic cellular level, and in doing so, we've revealed the remarkable, almost unbelievably profound impact of these microscopic cleanup crews, the proteostasis network and the autophagy lysosome system.
On what exactly?
On overall cellular health, the optimal function of our tissues, and ultimately on human longevity itself.
It's abundantly clear now that this isn't just, basic biology or some abstract scientific concept we learned in textbooks.
No, it feels much more fundamental.
It's a fundamental determinant of how our bodies age, how efficiently our biological systems operate throughout our lives, and whether we succumb to debilitating age-related diseases or manage to maintain vitality and resilience into advanced years. This is really about the core mechanisms of life's persistence.
And think about the sheer mind-boggling complexity and the elegant intelligence inherent in these microscopic autonomous processes.
That's staggering.
Cells autonomously manage their internal environment with incredible precision, constantly distinguishing between what needs repair, what needs shredding, and what needs to be fully recycled and rebuilt from scratch. It's a continuous, dynamic, and adaptive dance of maintenance, renewal, and molecular decision-making.
Happening constantly.
Happening billions of times a second in every single cell in your body right now.
So given the clear and compelling evidence we've meticulously discussed today, that boosting these cellular cleanup crews can extend a healthy lifespan and improve resilience across such a wide array of species, from tiny worms to complex mammals.
Yeah, the evidence seems strong.
What does this truly imply for future strategies in human health and medicine? This isn't just an academic question. It has immense practical implications, doesn't it?
It really does. Could a deeper, more refined understanding, and perhaps even the precise, targeted manipulation of these fundamental biological processes be the ultimate key?
The key to what?
To not just extending the human lifespan, maybe adding years to life, but unlocking true, robust health span in humans. Could this allow us to live not just longer, but healthier, more vibrant, and more functional lives, free from the burdens of chronic disease well into old age?
It's a question that continues to drive intense scientific inquiry and holds truly immense promise for the future of human well-being. A lot to think about.
Definitely a lot to think about.